running a little late. So today we're going to have 3 speakers, and the first is going to be James on the role of tryptophan metabolism in an animal model of inflammatory bowel disease. And we're tweeting along, the hashtag is ICUR14, hashtag, I think, S2B, sorry.
S2B. C, you mean.
S2C. Sorry.
Yes, C, you mean. Okay, cool. Thank you very much for coming to my presentation. Now, the thesis, title of my thesis has slightly changed, but today I'll be drawing the link between tryptophan metabolism and inflammatory bowel disease. So, just in case you're not that familiar with science, I'm going to give an outline of the introduction, where I'll be introducing the aspects of the thesis that you need to understand what I'm talking about. Then I will go through the aims and the hypotheses of my project before going through the methods. Following this, I will explain the results before I summarize and discuss the results. And finally, I will highlight the implications of my project. So a question. Which is a disease that affects 61,000 Australians right now, which costs Australia $2.7 billion per year, and which is being intensively researched right now in terms of the causes and the treatments? I've already reviewed the answer, just briefly.
Inflammatory bowel disease.
Yep, that is the correct answer, or IBD. Now, to define what IBD is, it is basically a chronic inflammation of the small and the large intestines. And normally, during IBD, you can see that the structure of the intestine is normal, which is uninflamed, and the intestines are designed to absorb nutrients from the intestine into the body. However, during IBD, the colon becomes inflamed, so that it becomes red and swollen, like so. And as you see later on, the structure of the intestine is lost, so that the person suffers from gastrointestinal symptoms such as cramping, abdominal pain, and eventually malnutrition. Now I would like to explain a bit about tryptophan. Tryptophan is an essential amino acid that you need to consume in the diet every day. Tryptophan is well known to be metabolized into serotonin, which is a neurotransmitter in the body, and melatonin, which regulates sleep and circadian rhythms. Previous studies on a high tryptophan diet have shown that animals who are fed a high tryptophan diet have increased responses to infection, so that they're able to clear the infection and survive, and as well as that there is decreased inflammation, especially in healing gastric ulcers and in many inflammatory diseases. So a high tryptophan diet has real potential to treat inflammatory diseases and inflammation. Most of the tryptophan that goes around the body is actually metabolized through the kynurenine pathway. And basically, IDO is the enzyme that catalyzes tryptophan into kynurenine. From kynurenine, it gets metabolized to many different molecules around the pathway, which are designed to suppress the immune response and inflammation. The molecule that I will talk about today is kynuric acid. And kynuric acid looks like this. Now, kynuric acid has been shown to block the many receptors in the nervous system, such as acetylcholine receptors and glutamic receptors. It has been shown that kynuric acid binds to the receptor GPR35, which is a metabolite sensing receptor that senses kynuric acid. Now, why is the link between kynuric acid and GPR35 important? Because previous studies have shown that GPR35 is expressed on many immune cells, such as monocytes of macrophages and iNKT cells. And that kynuric acid can bind to these receptors to suppress the secretion of many cytokines that promote inflammation. These include TNF-alpha, which promote massive inflammation, and IL-4, which promotes allergies and asthma. As well as that, GPR35 mutations in GPR35 have been associated with IBD, as these mutations render the GPR35 receptor less functional, and hence the person becomes more susceptible to the inflammation associated with IBD. Now, I'm drawing the link between tryptophan, chymeric acid, GPR35, and IBD because the roles of tryptophan, chymeric acid, and GPR35 in IBD, as well as the mechanisms linking these two together, are relatively unknown. And this is the thing that I will investigate. So the aim of my project is to determine whether a high tryptophan diet, chymeric acid, and GPR35 can affect the severity of an animal model of IBD. I hypothesized that a high tryptophan diet, chymeric acid, and GPR35 can increase the severity of an animal model of IBD. Now, what is the animal model that I use for this project? I use DSS colitis, which is an animal model of IBD. Basically, how it works is that dextrosodium sulfate is a long molecule that you can dissolve in water. Now, the mice drink this water, and they also ingest the DSS molecule. This goes on for 7 days. During that time, you can see that the colon becomes from this, in the normal state, to this, which is massively damaged with lots of inflammatory cell infiltration and damage of the colon. Now, this is a piece of the colon that occurs when the mouse ingests DSS for 7 days. And this resembles that of inflammatory bowel disease. Now, in terms of my experiment, most of the experiments I did, I used 6 to 10 week old female C mice. And basically how it worked is I put the mice under a controlled diet before I put the mice under a high tryptophan diet or chymeric acid in drinking water for a number of days. During this time, after this time, I started DSS colitis, and I monitored them for 7 days in terms of weight and clinical scoring, which was basically checked for blood in the feces, checked for blood around the anus, and also the fecal consistency. The endpoint was around that time, and we did endpoint experiments. In terms of the GPR35 knockout experiment, we used male mice, which were either wild type, which had all functional genes, or GPR35 knockout mice, which did not have a functional GPR35, to see if GPR35 might contribute to the development of DSS colitis. And now I will present my results. Firstly, we found that mice that were on a high tryptophan diet for 7 days had a reduced severity of DSS colitis. Normally, during DSS colitis, because you're replicating IBD, the mice that were on a control diet lose a massive amount of weight, resembling that of IBD, as the mice do not consume or drink that much food or water. However, we see that the mice that were on a high tryptophan diet before had a reduced weight loss. As well as that, in terms of DSS colitis, the symptoms of DSS colitis in the control mice worsen over time. However, in the tryptophan mice, the severity of DSS colitis is less. We put these mice under a high tryptophan diet for different periods of time, and we found that mice that were on a tryptophan diet for 3 or 14 days did not have a reduced severity of DSS colitis. In fact, as you can see here, the mice on the high tryptophan diet have actually lost more weight compared to the control mice. And as well as that, the increased severity of the symptoms in both 3 days and 14 days was the same in both the control and the tryptophan diets. We then looked at the histology of colons to try and see if we can confirm the monitoring results. What you can see here in this colon is that you have a lot of massive inflammatory cell infiltration, and basically the structure that you see over here with its crypts or pits and the epithelium is virtually all lost in a field of inflammatory cells. However, the structure of the high tryptophan diet under DSS colitis conditions remain relatively intact, besides a few inflammatory cells. what you can see here is we scored the columns based on the damage of the structure and inflammatory cell infiltration. And what we can see here is that the physiological score in the high tryptophan diets was less than the control diets. We then checked that on the 3 and 14-day tryptophan diets, but, oops, sorry, that's the wrong one. We then checked the histology of the tryptophan diets, and what we can see here is that the damage of the columns was relatively the same, with the same amount of damage. And what we can see here for, in terms of histology, is that the histological score between these two groups of mice was the same between the 3 and 14-day tryptophan diets, showing that you need mice that were on the high tryptophan diet for 7 days only reduces the severity of the SS colitis. We then checked chymeric acid and see if putting mice on chymeric acid for 7 days could reduce the severity of the SS colitis. Unfortunately, that was not the case. As you can see here in the rate change, the mice on water and chymeric acid under SS colitis conditions lost a similar amount of weight. And although the chymeric acid mice maintain the severity of symptoms over the first 4 days, overall the difference between the water controls and the chymeric acid controls was not significantly different, as the chymeric acid controls continue to increase in the severity of symptoms. Looking at the histology of the columns, we can see that the amount of damage in the water and the chymer groups under SS colitis conditions remain around the same. And despite the fact that the mice on the chymeric acid tend to have low histological scores compared to the water controls, this difference was not significant. These results show that mice on the chymeric acid for 7 days do not help to reduce severity of the SS colitis. So, looking at this result, we want to find out whether GPR35, the receptor for chymeric acid, has any effect on SS colitis. Now, the results I will present are only preliminary, as we only had 4 mice per group, and we didn't really have a control group. But looking from these graphs, the rate change between these two groups was similar. But unexpectedly, the GPR35 knockout mice had less severe symptoms under lower clinical scoring compared with the wild-type mice. Looking at colony, it can be seen that normally wild-type mice on the SS colitis conditions have shortened colons, which are more inflamed, and they also have inflamed sepums. On the other hand, for the knockout mice, the colon length was relatively maintained, and as well as that, the colons were relatively more normal than those of the wild types. Looking at the colon length, it can be seen that the mice on the knockout mice had relatively longer colon lengths compared with the wild type mice. If we look at the histology of the colons, it can be seen that the wild type mice had lost the crypts and the structure with a blanket of inflammatory cells and some loss in the epithelium. On the other hand, other than some inflammatory cells being infiltrated on the colon, the structure of the colon with the crypts are relatively intact. Looking at the histological scores, the GPR35 knockout mice had relatively lower histological scores than the wild-type mice. So to summarize, we found firstly that mice that were on a high tryptophan diet for 7 days had a reduced severity of the SS colitis, which was in agreement with the fact that tryptophan and its metabolites are able to suppress the inflammation. However, chymeric acid was not a molecule suppressing the inflammation, as it did not reduce the severity of the SS colitis. Now, this did not sit well with the previous literature, as I previously explained that chymeric acid suppresses the secretion of pro-inflammatory cytokines that promote inflammation. One case might be that the dose or the route of administration of chymeric acid might need to be optimized so that all of the chymeric acid enters the colon and exerts an effect, as well as that another tryptophan metabolite in the chimerin pathway might be involved in this process. And lastly, surprisingly, we found that knocking out GPR35 reduced the severity of the SS colitis. But as I have explained, the results I have presented were only preliminary, and there are many limitations such as only 4 mice per group, and there was no control group to compare the DSS mice with the control mice. The implications for my project are as follows. Firstly, by undergoing this project, we have a better understanding of the role of tryptophan, tartaric acid, and GPR35 in IBD. And at the same time, we found that tryptophan diets would suppress the development of DSS collides. So an implication of this might be that by modifying the diet or giving tryptophan supplements, we may help to alleviate the symptoms of IBD. Well, I'd like to thank the following people who have helped me in my project. Thank you.
So now we might have a few minutes of questions, if anyone has any questions they'd like to ask. What's the some future work that you recommend to further this research?
Yeah, sure thing. So basically, we need to reconfirm the 7-day tryptophan diet just to confirm our results, because it was a bit strange to me that mice that were on tryptophan diets for 3 and 14 days had no effect. So we want to confirm the 7-day tryptophan diet again. As well as that, we definitely need to run the GPR35 knockout experiment again, because of the small sample size. And the problem with that is that GPR35 knockout mice tend to breed a bit less compared to the wild type mice. So we're hopeful that we can gather up enough GPR35 knockout mice to run an experiment with a bigger sample size, and obviously with controls. As well as that, I'd like to compare the ICUR expression, which is basically the enzyme that is involved in the kynurenine pathway, and see if the ICUR expression is different between mice on tryptophan diets over time. And obviously, lastly, I like to run a different animal model of IBD, because besides DSS collides, there are actually many different animal models of IBD, which target different areas of the immune system and inflammation. So that's where we like to head to next. Yep.
I think I noticed in one of your early slides that one series of experiments was done with male mice and another series with female. Is there a reason for that?
Yes. Ultimately, we'd like to actually use the same genders for both of the experiments. In terms of the GPR35 knockout experiment, this came out of the blue because initially I thought, because we didn't have enough GPR35 knockout mice, that we wouldn't be able to run that experiment. But luckily we had GPR35 knockout mice, maybe within August. But the problem was the only GPR35 knockout mice that were available were males. So to cover these results of the GPR35 knockout experiment, we had to use male mice. Obviously, we would use female mice, because that way the results of the GPR35 knockout experiment and the other experiments would be dead comparable. But we had to use male mice because they were the only GPR35 knockout mice available at the time.
So will you go back and repeat it with female mice?
If we have enough female mice. As I highlighted early on, reading the GPR35 knockout mice was a bit difficult, basically because they tend to produce less. Tend to produce less offspring compared to normal mice. But that is optimally where we like to head to next.
Thanks. If we have any more questions, we might bring it up at the end.
Yep,
We'll go to the next speaker then. So give it another call. Next lecture will be Hakan Tarakci speak to us about zebrafish models for myopathies caused by mutations in TPM3. Just to let our speakers know, I have signs here. I'll tap my leg and I'll put them up for 5 minutes and 2 minutes left.
Thank you, Josh, and good morning, everyone. As I'm sure all of you are aware, the basic function of muscle is to generate force. And there are 3 kinds of muscle that collectively make up approximately half of our body weight. And these are skeletal muscle, cardiac muscle, and smooth muscle. And for the purpose of this presentation, I'm going to be focusing on skeletal muscle. Now, when we think of muscle, skeletal muscle is what comes to mind first, and it's the muscle that's voluntarily controlled and helps us move and maintain our posture. And it's often sometimes referred to as striated muscle due to the striking appearance under a microscope. Now, skeletal muscle is almost always attached to bone and is made up of many muscle fibers. And each muscle fiber is a collection of myofibrils. And these myofibrils are made up of end-to-end tandem repeats called sarcomeres. These sarcomeres are the functional units of skeletal muscle, and they're composed of long fibrous proteins that slide past each other when the muscle contracts and relaxes. These are made up of thick filaments, which contain myosin, and thin filaments, which contain actin. Now, the binding of actin and myosin with energy provided from ATP allows for muscle contraction and movement. Zooming into one of these thin filaments, we can see the ordered arrangement of proteins, and my honours project has been focusing on tropomyosin, which is shown here in grey. So tropomyosin is a family of highly conserved actin-binding proteins, and they play a broad number of roles in cell responses, such as regulating cell shape, motility, and involving cell pathways. But interestingly for me, in muscle, it plays a role in regulating muscle contractions and also acts as stabilizer thin filaments. So tropomyosin at rest lies along the actin filaments and covers the myosin binding sites. And then through the release of calcium, we get the shift in the position of tropomyosin to expose the myosin binding sites. And with the myosin binding sites now exposed, actin can now bind to myosin, which allows for muscle contraction. In humans, there are 4 homologous forms of tropomyosin that evolved through genome mutations. And of these, I'm focusing on tropomyosin 3, which is expressed in type 1 slow muscle fibers and also the heart. And so type 1 slow muscle fibers are the ones important for maintaining posture and contract for long periods of time, as opposed to type 2 fast muscle fibers, which is for generating large amounts of force for short periods of time. And diseases of muscle are termed myopathies, and in the literature there's currently over 600 different kinds of myopathies described, and they range from mild muscle weakness during adulthood to fatal forms of neonatal onset. And it's known that mutations in TPM3 can cause myopathies, and we know that's associated with membrane myopathy, which is characterized by the presence of these protein aggregates called membrane bodies that cause muscle weakness. Mutations in TPM3 have also been shown to be associated with congenital fiber type disproportion. We will see decreases in size in the type 1 muscle fibers compared to type 2. And mutations in TPM3 have been found in families suffering from capnopathy as well, which is characterized by the presence of these cap-like structures within the muscle. There are currently 21 mutations known in TPM3, and my honours project is focused on 4, M9R, L100M, R168C, and R168H. But for the purpose of the presentation, I'll be focusing on giving the results on M9R, which is associated with membrane myopathy in humans. It's the first mutation that was found in TPM3. It's also the most well studied and known, and also it results in one of the most severe forms of myopathies in humans. Which brings me to the aim of my project, which is to characterize and investigate the mechanism of disease of myopathies caused by mutations in TPM3 using zebrafish as a model organism. So using zebrafish has many advantages. Its optical transparency during development allows for live in vivo imaging. In addition, they have a very fast external development and produce high numbers of offspring, where you typically get approximately 100 to 200 eggs in a single clutch. They're also vertebrates, and the genes and mechanisms by which muscle develops is highly conserved between zebrafish and humans. And we also know that they're excellent for screening, just because they produce high numbers of offspring, and they're very easy to genetically manipulate. Now, in order to achieve my aims, I've gone about screening and identifying transgenic zebrafish and also generating mosaic fish. And these feel a bit different in that transgenic fish, my transgenic fish, are expressing tropomyosin 3 within muscle cells. And the mosaic fish, you can see individual breed fibers. So it's not in all muscle cells, but only in a subset. And so using both the transgenic and mosaic fish, I've gone about analysing the structure of the muscle using 2 techniques, primarily light confocal imaging and antibody staining. And in addition to this, I've analysed the swing behaviour using 2 independent swimming assays. So confocal microscopy allows for high resolution images, and the fact that zebrafish are optically transparent makes it a lot easier as well. And this has the potential to generate three-dimensional images as well. So having a look at the M9R fish at 48 hours, we see a collection of wavy fibers located throughout the muscle, as indicated with white arrows. And this persists at 72 hours as well, but at a much lower frequency. And so we can see individual fibers that are striated, so they have TPM3 incorporated into the sarcomeres. However, the integrity of the muscle fiber seems to be disrupted, resulting in wavy fiber. Additionally, antibody staining shows that occasionally you do get some striated fibers, but also what I've been told is some aggregates forming, which may be due to the presence of too much tropomyosin. Now, the swimming assays that I've used, the first one is the touch and birth response assay, which is looking at the embryo's response to tactile stimulation. And this was measured at 2 days, 2-day-old fish. And here we have the typical response from a fish, which is just swimming the other direction as fast as it can. And the reporting system just measures the embryo's response. And then we can use software such as Zebrafish to get an output. And it measures many variables and parameters, which we can then quantify and measure accurately the fish's motion. So we know that force is proportional to acceleration, and so I've had a look at the maximum acceleration of the fish as a great indicator of the maximum force that the fish can generate. And interestingly, we find no significant difference within the M9R transgenic fish and the appropriate controls. And this was looking at 3 independent clutches. I've also had a look at the maximum speed that the fish travel at, and again, there was no significant difference between the controls and transgenic fish. Now, the second assay that I've used to have a look at swimming behaviour is the Zebrabox analysis. And Zebrabox is a reporting system which allows you to analyse 96 zebrafish simultaneously. And again, you can use software such as Zebrafish to quantify and measure the motion of the fish as well. So here we have a Zebrabox reporting system which shows the potential to record 96 fish simultaneously. Has a great advantage because it's you're looking at so many fish just at once as opposed to the touch and go response assay where you're analysing one fish at a time. So a typical response we get from Zebrabox, we can see in black, green is where the fish was stationary, green is where the fish is making small movements, and the red lines mark where the fish made large movements. And so once again, using Zebrafish, we've got one of the parameters I looked at was the movement count, which counts how many times the fish has moved over a period of 10 minutes. And there was no significant difference again between M9R transgenic fish and the appropriate controls. And I've also had a look at the total distance that the fish cover. And although there was a slight increase in the M9R transgenic fish, there was no significant difference between the controls as well. So that shows that there's no significant difference in the locomotion at 6 days, and the touch and birth response assay showed that there was no significant difference in the acceleration and maximum speed of the fish. So I'm finalising my honours project now, and I'm currently processing samples for electron microscopy just to get greater detail on organisation of filaments, which we wouldn't be able to get at that resolution from focal microscopy. Further analysis of the wave fibers to sort of characterise them, and possibly looking at localization of other proteins using antibody staining. In addition, I'm currently constructing DNA sequences that can be used to generate double transgenic fish. And so my goal is to have a look at actin and legal as green, and look at tropomyosin and legal as red. So we can just see an example of a double transgenic fish. It's a lot more powerful as opposed to looking at where one protein is, you can look at where both of them are at the same time. So currently there's no cure or treatment for myopathies, and there are measures to increase the quality of life, such as giving antibiotics to treat chest infections and physiotherapy, but all of these are quite limited. And so this is the first study to model myopathies using zebrafish as a model organism caused by TPM3 mutations. Now, identifying a mechanism can therefore aid in therapies, and establishment of a successful model opened up the opportunity for drug screening and hence the development of a treatment. So I'd like to thank personally some people that I couldn't have done this without. So the Bryson Richardson Lab, my supervisor, Dr. Rob Bryson Richardson, Caitlin Tamar, Biomedical, Nick and Jonathan, also the Cave Lab and the Australian Medicine Institute for the Health, and also Fish for the housing of the fish, but also ICUR for organising this event.
So does anyone have any questions?
just have a question. You noticed that there was no change in the movement between the wild type and the mutated fish. Would a reason probably be because the zebrafish don't have that much muscle to move around it with, and so there wouldn't be that much of an effect in movement compared to using a rat or a mouse, for instance?
Having looked at fish, everything that's green is muscle. So the majority of the fish is muscle. No significant difference just means that there was no little difference between it. It could be that at a later stage it might be showing up. Because I was looking at fish at 2 and 6 days. It might be just a later onset, so it might be happening at a later stage.
So how long can these fish live for?
Typically over a year, but they've reached maturity at about 3 months.
3 months, okay. Yeah. So maybe you probably be investigating older fish, say 3 months and beyond, and see if there's an effect.
I think it's going to be easier.
What about some of the other mutations that we've looked at?
I've studied MIR in greater detail in the other mutations, but again, the other ones, the mosaic fish are showing some abnormal fibers similar to what we saw in MIR, but the confocal imaging doesn't have the wavy phenotype, and that just might be because the MIR is an important domain, which is important for why it's active and also for head-to-tail performance. So there's no significant effect in the other mutations, but this is a more dramatic one.
Well, I might ask a question. So, what would you have been expecting to get, I guess? What was your hypothesis when you get this experiment?
Well, we're expecting to see what we see in mutations, which is the number of bodies that we showed on the earlier slides. So those are the protein aggregates that form as a result of immune form being expressed. And so we haven't bumped into any of the bodies, but the wavy fibers have been surprised because it's not really something that we expect to see. But again, it is a defect, so it's about characterizing that and working out what aspects of that can be transferred into human patients and then using that as a model.
Well, we might leave it there then. If anyone has any questions later, maybe you can have a round of applause. And next we have Ingrid Augusto to speak to us about localizing the regulator protein RabGap TBC1D4 inside the
autophagy.
pathway.
Good morning, everyone. I am Ingrid Augusto, and I will present about the research project that I'm developing in Monash, and it's about the localization of the protein RabGap TBC1D4 inside the autophagy. I will give you a background about the autophagy and the proteins Rab and Gap, and stay to the end of this project and explain a little about the two methods that I'm using. It's the Immunology and Immunology, and discuss about the partial results that I took, and give the future directions that we will be taking this project. First, what is autophagy? Well, it can be basically defined as a degradation pattern in which the intercellular material and organelles and cytosolic material will be delivered inside the lumen of the lysosome to be degraded. And why these organelles should be degraded? Because the autophagy will keep the homeostasis of the cell by cleaning the cytosol from old organelles or damaged organelles or aggregate proteins that are elements that can disturb the cell behavior. And also, autophagy will give recycling of nutrients through after degradation. These nutrients can be reused, and this will help the cell in stress conditions like nutrient starvation, and will guarantee the cell survival. Also, autophagy, it's important in many physiological processes like growing cells and diseases like neurodegenerative organisms. the mechanism of autophagy pathway. There are two types of autophagy, but I'll explain the autophagy, that is the main one. And it starts, the first step is the induction. Here, autophagy. There we will start at phagophore assembly site. It's one specific site in the cell where many proteins involved in vessel formation will be recruited to start the formation of a small double member compartment, and that will this establishes the nucleation and elongation of this double member, and then we will involve the material that will be degraded, and many proteins are recruited here to be from the autophagosome. There is a double member vessel that will be transformed into the lysosome, and then here's the fusion, where this material will be delivered in the lumen, and here are many hydrolyzers enzymes to degrade this material. All this mechanism is very well regulated, and one of the proteins responsible for regulating this process is the RAB proteins that do part of a larger family of small GPRs, and its function is mainly coordinate the membrane traffic inside the cell. It's one example of the diversity of RAB proteins that exist. In humans, there are more than 6 types. And these proteins are distributed in all the cell, and there are specific RABs located in specific organelles or membranes responsible in different pathways in the cell. And these RAB proteins are regulated by these two regulator enzymes. One is DEF, or guanine nucleotide exchange factor, that will activate this RAB protein. So we have the RAB protein in your inactive form. It's attached with the GDP molecule, and then the DEF will activate by chain switching the GDP molecule to GDP, and in your active form, the GDP bound form, the RAB protein will then bind to other proteins called effectors that will do the downstream function of this of the RAB. And the interesting point is that although the name is activating protein, this GAP protein will inactivate the RAB by hydrolyzing the GDP molecule and come back to the GDP bound form. The GAP proteins are specific as well to specific RABs. And it's important not only to inactivate the protein, but also regulate where and when the RAB protein will act. So we have here specifically the TBC1D4, so a kind of GAP protein that can activate the RABs, and it's well known in the regulation of glucose transport. But recently, in 2012, was identified by 2 different authors inside the autophagy pathway. Monash identified between 11 RabGaps that inhibit the autophagy, and Popovic identified proteomic this protein in the autophagy. But we don't have too much information about this, that this gap is present in the autophagy. And then the aim of this project is localize and provide more information, morphological information about the function of the gap TBC1D4. So I'm using 2 methods. One is Immunology and the other one fluorescence. It's basically the same idea. We use one antibody specific for TBC1D4. In the gold level, we have a gold partner with a protein A that will bind in the antibody. And in the fluorescence method, we have, instead of a gold partner, we have a fluorophore that will emit fluorescence. And the immunology level is analyzing in electron microscope, and the fluorescence in a focal microscope. We use derivative cells because this type of cells has a natural highest expression of TBC1D4. And we did 2 groups, one with induction of autophagy using vancomycin and bacteromycin, the other one in control without this induction. To analyze these samples in microscope, we need perpetual autophagy to provide the structure for the cell being coated, and also to prevent change in the structure of the cell. like formation of artifacts. So the first step is the fixation, and then we invert the gelatin to provide structure to the protein. And this gelatin is set in many blocks, small blocks, and then we do a sucrose filtration, because sucrose prevents the formation of ICUR itself. And then we freeze with liquid nitrogen. Then, using this equipment, ultramicroscope, where it's pumped every time, all time, with all the liquid nitrogen to keep the sample in negative temperatures, in 115 or 120. And it will trim it. There is like a draw on the square in the block, using a knife of diamond. And then the part section is the part that require more technical skills, because we do the equipment we section. And we form like a ribbon, and I need to put this ribbon down, the knife, using a stick with a elastic tip, and then using a specific loop with sucrose, we do the cap and put it in a grids, metal grids, or in cover sheet. To do the gold level, the section is 6 nanometers, and we tested different concentrations of antibody BCIR4 to set up what could be better, and we scan it and then observe the electron process. The difference to the fluorescence is that the section is 200 nanometers, that is easy to cut and analyze, and we use the fluoroalexan for 88 to bind the antibody and the host to stain the nucleus to be easy to identify itself. And we test only high concentrations of antibody BCIR4 because of the results from the Immunology lab. Then we observe comforting the process. So the results from the first Immunology lab that I did is that here is what I come across the image, and we can observe just a few gold particles, like 2 here. And its image is from a high concentration of antibody, but it's still a few gold particles inside. And also, because of the first time that I did this, like I explained, this technique that required good technical skills, I also found many technical issues, like this one, the bad image, with overlapping of sections that result the interpretation. And the summary from the first Immunology, I have few gold particles in high concentration, and I asked if it's because of technical issues or could be some problem with the antibody. So I did again the price section after the price, and I got good morphology, less technical issues like here the membrane, and one huge mitochondria. It's better to analyze the cells. But the problem of few gold particles is still. I have just not too much information from that, and I can't identify also vesicle autophagy structures in this sample. So even with less technical issues, the low level efficiency from the antibody is still, and I didn't identify the autophagy structure. So the question that is still could be some problem with antibody. And we decided to do the fluorescence because it's easy to cut and to know if the antibody is working or not as well, to test in a second technique. And the results is the control. We can see that the antibody is working. It's binding here in both and in the control and in the autophagy induction sample. But I expect that the autophagy sample with induction was brighter than the control. But if you look, the pattern of brightening and presence of control dots is the same. It's very similar. So the antibody is binding, but the pattern is the same. And I cannot confirm that it's autophagy. And I also asked him if it's not the one problem in the autophagy induction of the beginning of the samples. So what we do next is test using a positive control, using the protein S3, that is an autophagy marker. protein very well known that is participating in the autophagy process, and we do a double labeling with antibody for FC3 and TBC1D4 to see if these blood clones are in autophagy or not. And then also to the Immunology technique, we will try different antibody for TBC1D4, maybe to increase the efficiency of labeling and try get the answers about the localization of this protein. I want to acknowledge the member of biology and molecular microscopy lab, and also the professor Dr. George Lamb and the lab members Adam and Bill for assisting on the project. And my sponsor that is the Brazilian government and the program of Ciências Sem Fronteiras that make me the opportunity to study Monash and do this research.
Anybody has any questions?
Yeah. Okay. Could you go back to the immunofluorescence images? Yeah. Over there. From my perspective, I can see that there is slightly more green fluorescence in the autophagy induction than in the control cells. Did you try and quantify the amount of green fluorescence in both of these images to give you a quantitative amount of fluorescence?
No, I didn't. The quantification was it's more to look if the network was working or not. And also, if you look at the magnification and the difference between the control and the induction.
Right.
Yeah.
So which magnification is closer? Which had a higher magnification?
It's the result of induction.
Oh, okay. With the
autophagy. This is like more. But the pattern of the presence of the right components is the same. It's very similar in the cytosol, and yeah, it's very similar. I cannot say that this was.
Do you see expression of your protein in cells that are not undergo autophagy?
Cells.
So you struggled to find in the end because you didn't have autophagic vesicles, but do you know where you would expect the protein to be when autophagy isn't?
In the autophagy. It's expected in the beginning with autophagy information. Here, because was in the literature, they described that the TBC1D4 is binding to ATG 101, that is one protein present here. Information of the autophagy. So problem should be found in the beginning of the formation.
So the first one is just your antibody could be working, but you just haven't found
This because electron microscopy is different technique. So for this we do the fluorescence to give more overview about this.
You chose dendritic cells, and can you tell me again the reason for that? Sorry, it's dendritic cells that you chose. I think one of the slides that you chose was
About different
no, back. No. Yes, primary dendritic cells. So at the highest expression, and is that typically what's used to study
the TBC1D4 is first described in insulin glucose response, and this is using adipocyte cells. But to try to find in autophagy the cell of the meat because it has a high expression, we thought to use first the adipocytes, but Professor Dr. said people actually try this one because of the high expression. Another possibility to try and find localize this is also the overexpression. Use one cell with overexpression of this protein. But this is a good possibility because it can do a mislocalization. So we use this because natural has a high expression. It's the first part.
Does anyone have any more questions for Ingrid or any of the other speakers?
I have a question. I'm curious about where you got your Hugh Walpole quotation at the end. How did you come across Hugh Walpole?
I searched in the internet about some phrase that could express what happened in the project. That I had too much issues and did some mistakes during the experiment, and rather we do this first. It's still looking to try the tool, to try to find the real for the science. Just to think about it.
Yeah, really nice. Good addition. Thank you.